high burden. Currently, there are no vaccine or cure for HIV, and only about half of the 39 million HIV infected people have access to antiretroviral treatment, ART. Furthermore, these people must rely on ART to suppress their virus levels for life. Interruption of treatment will lead to virus rebound back to pre-ART level, and subsequently lead to disease progression and onward transmission. So in this graph, we can see suppression of HIV virus level upon the initiation of ART, and then why the cessation of ART will lead to virus rebound back to pre-ART level. So surprisingly, a small subset of or group of people with HIV develop broadly neutralizing antibodies, which are known as BNAbs. They do so over the course of 2 to 3 years of untreated infection, as compared to the rest of the population who develop narrow spectrum neutralizing antibodies. So if you look at this pie chart, we can see the distribution of neutralizing antibodies among the global HIV population according to how many HIV variants these antibodies can neutralize. Although about 1 to 2% of HIV population, the region in red, develops broad and protein neutralizing antibodies, no HIV specific antibody has 100% breadth or coverage against all HIV variants. And that is why individuals who develop broad neutralizing antibodies cannot clear their own viruses, because some of their viruses will escape antibody responses. So as we may know, HIV mutates rapidly, and because of mutation and other escape mechanisms, which we talk about later, there will be quasi-species in every infected individual. Therefore, in any HIV-infected individual who develop broadly neutralizing antibodies or BNAbs, a resistant strain will emerge that will escape antibody responses as compared to sensitive strains. So because of broadly neutralizing antibodies' ability to neutralize a wide range of HIV variant, a couple of broadly neutralizing antibodies are being isolated and passively administered in animals and human studies to prevent HIV acquisition. So this prevention is strictly limited to virus strains that are sensitive to a particular broadly neutralizing antibody. So in 2 recently concluded randomized clinical trials designed to investigate whether passive administration or infusion of VRC01, which is a broadly neutralizing antibody, whether it can prevent HIV acquisition. So in this study, which I think was conducted in South Africa, over 6,000 individuals who received or did not receive VRC01 every 8 weeks were followed up for up to 80 weeks and monitored for HIV infection every 4 weeks. And at the end of this study, the infection of VRC01 was now found to prevent overall HIV acquisition as compared to placebo. However, against its pre-specified sensitive viruses, VRC01 was found to be 75% efficacious. So the table on the right here shows the incident rates for sensitive, which are the one here, as compared to resistant viruses among people who received or did not receive VRC01. For resistant viruses, the rate of new infection in both placebo and VRC01 groups were similar. However, for sensitive viruses, the rate of new infection were lower in VRC01 as compared to placebo group. So these studies serve as a proof of concept that broadly neutralizing antibodies may prevent acquisition of sensitive viruses. So furthermore, broadly neutralizing antibodies, when administered, can momentarily suppress virus levels. So in this study, HIV-infected individuals were infused with 3BNC117 antibody at two different concentrations on day 0, at 10 and at 30 milligrams of antibody per body weight in kilogram. We can see that the HIV virus level, HIV RNA level or virus level dropping for at least 21 days before going up to pre-infusion level. The rebound back to pre-infusion level is likely because of virus escape from the antibodies or because of antibody decay over time, or it could be because of both factors. In another study comparing the time it takes for HIV to rebound in people with HIV who paused their ART, the infusion of broadly neutralizing antibodies, and in this study, 3BNC117 and 101074, the infusion of broadly neutralizing antibodies was found to delay virus rebound for up to 44 weeks as compared to 8 weeks for people with HIV who did not receive antibodies. So approaches to induce broadly neutralizing antibodies with vaccine regimen have only worked in animals, but not human so far. One of these approaches involved um, isolating broadly neutralizing antibodies from HIV individual who naturally develop them, and then tracing the HIV antigen, in this case, HIV envelope, that this individual co-evolved with to reflect affinity maturation. So once these envelope structures are uncovered, all infected animals may then be vaccinated in a sequential manner, starting with the envelope structure that will select the broadly neutralizing antibody precursor, followed by the intermediate specific envelope, and finally the native trimeric envelope. So this lineage tracing approach has been used to design immunogen that induce broadly neutralizing antibodies in animals, but we have not been able to successfully induce broadly neutralizing antibodies in humans. So in addition to immunogen or antigen design, attempts to improve HIV vaccine also include comparing the immunogenicity of different adjuvants. That is, comparing the ability of different adjuvants to enhance antibody levels in response to HIV envelope antigen in the vaccine regimen. So in animal studies, formulations of HIV envelope and freehand complete adjuvant, or FCA, derived from Mycobacterium tuberculosis, have been used to boost antibody responses as part of a vaccine regimen. So in this study, animals were immunized 3 times over 16 weeks with HIV envelope, with or without adjuvant. And as compared to other adjuvants, animals who received freehand adjuvant, the same adjuvant that is derived from Mycobacterium tuberculosis, and HIV envelope formulation had higher levels of antibodies and avidity or binding at the end of the study. We can see here the group 1 here had threefold antigen, and group 2 had one-fold antigen. And the envelope antigen did not affect the antibody responses in these animals. So although the use of freehand complete adjuvant, FCA, derived from Mycobacterium tuberculosis, is banned in humans, Mycobacterium tuberculosis, or MTB, is the most common opportunistic infection in people with HIV and is reported to be involved in heterologous immunity. So heterologous immunity describe an event whereby a first infection influences the immunological responses to a second non-identical pathogen. So Mycobacterium tuberculosis-driven heterologous immunity could be due to the development of cross-reactive B cells responding to an epitope present on tuberculosis and the second non-identical pathogen, in this case, HIV. Or it could be due to the ability of Mycobacterium tuberculosis to epigenetically remodel monocytes to improve monocyte readiness or activation status during a second or concurrent infection like HIV. So this improved readiness may lead to excess secretion of cytokines that may transiently activate B and T cells that are specific to the second pathogen. Additionally, the presence of Mycobacterium tuberculosis disease in people living with HIV has been shown to increase determinants or factors that drive the generation of broad and potent neutralizing antibodies in people with HIV. So some of these determinants include the formation of granulomas, which is the hallmark of tuberculosis disease in the lungs. So this granuloma contains organized immune cells and may increase the interaction of between B and T cells. Secondly, the increase in HIV replication increases the overall viral load and diversity, which may improve the quantity and quality of HIV antigen required to generate broad and potent antibodies. And then the third factor is that tuberculosis disease increases cytokines that may be involved in B cell development. So taken all together, we proposed a model which sought to investigate how active tuberculosis perturb HIV specific antibody responses. So in this model, we proposed that the presence of tuberculosis in people with HIV may lead to epigenetic remodeling of monocytes and macrophages through the pattern recognition, through the engagement of the pattern recognition receptor. And this remodel macrophage may secrete high level of cytokines that may enhance HIV-specific B cell survival and also promote HIV-specific T follicular helper cells that may altogether come together to enhance, to lead to the generation of Jamina, to lead to the establishment of Jamina center. And then these selected Jamina center B cells may differentiate into plasma cells or antibody secreting cells, which would then secrete broad and potent neutralizing antibodies. So on the contrary, and in the absence of tuberculosis, the elicitation of antibody responses may be weak due to the low level of secreted cytokines, insufficient to drive a Jamina center reaction. In the absence of T follicular helper cells, HIV-specific B cells may differentiate into antibody secreting cells that may only secrete non-neutralizing antibodies. So we leverage on the high burden of HIV and active tuberculosis to recruit participants from 6 countries across 3 continents. Before treatment, 52 people with HIV were screened for active tuberculosis. And in this study, we confirmed active tuberculosis with positive test results to microbial culture, sputum smear microscopy, and Gen expert assay. 15 participants had active tuberculosis, and 37 participants, that is to say people with HIV, had no active tuberculosis. We did not assess if these individuals had latent tuberculosis. So plasma samples from these individuals were obtained, and all individuals started HIV and all tuberculosis treatment. So after about 6 months of treatment, 41 individuals returned, while 11 individuals dropped out. 10 people with HIV who initially had uh, tuberculosis now tested negative to tuberculosis and were assigned people with HIV and treated tuberculosis. And then in the other group, in the other group, we have 9 of these individuals who develop tuberculosis, and 22 individuals remain without active tuberculosis. So in summary, we have 2 groups before any treatment, and we have 3 groups after treatment. So this study is cross-sectional, and we compare the personal characteristics of participants before and after antiretroviral treatment, or ART. So before ART, age, gender, and virus levels were similar among the 2 groups. However, the CD4 count was higher in people with HIV and active tuberculosis as compared to people with HIV, no tuberculosis. At follow-up or post-ART, age, gender, virus level, a number of individuals who reach suppressed virus levels and CD4 count were similar among people with HIV treated tuberculosis, people with HIV active tuberculosis, and people with HIV no tuberculosis. However, the duration of ART was longer in people with HIV treated tuberculosis and people with HIV no tuberculosis as compared to people with HIV active tuberculosis. The p-value is there. So going back to our model, the first thing we did was to examine the functional qualities of HIV-specific antibodies. Essentially, we wanted to know whether antibodies in people with HIV and active tuberculosis, as compared to those in people with HIV and no tuberculosis, are broader and more potent against different HIV variants. So we focused on 2 functions of specific antibodies. And one is neutralization, which involves the antibodies binding cell-free variants with the FAB portion to prevent HIV entry into a target cell. The second aspect we focused on was antibody-dependent cellular cytotoxicity, or ADCC. And in ADCC, specific antibodies bind, express HIV envelope on infected cells. And then natural killer cells then engage the FC portion of these bound antibody through the FC receptor on the natural killer cell, or CD16. And then once this complex is formed, the natural killer cells release molecules, precisely granzyme and perforin, to clear this infected cell. So furthermore, we selected a panel of 12 viruses that we know are difficult to neutralize. So these difficult to neutralize or tier 2 viruses are drawn down from over 200 globally sampled viruses. And responses against these 12 viruses are similar to responses to these over 200 globally sampled viruses from which these 12 viruses originated from. So finally, this panel is used for the standard assessment of neutralizing antibodies in the field of HIV. So to assess antibody neutralization activity, we adopted a cell reporter assay. And in this assay, the TZMBL cells have been engineered to express receptor CD4 and co-receptor CCR5 and then CSCR4, all of which are required for HIV entry. Furthermore, these TZMBL cells have a luciferase reporter gene that requires HIV TAT protein for expression. HIV TAT protein is one of the earliest protein expressed upon HIV entry into target cells. So in a situation whereby the plasma or the antibody cannot neutralize these cell-free variants, HIV will enter the cell, produce TAT protein, and then this protein we then transcribe the luciferase gene. And with the addition of stimulator, the infected cells will produce luciferase. Luciferase is a light-producing enzyme naturally found in fireflies. Conversely, a complete neutralization of these cell-free variants would prevent virus entry and luciferase expression. So remember that we are using 12 difficult to neutralize HIV variants in this study. So we estimated the neutralization breadth and potency score, or BP score, for each participant. The neutralization BP score tells us two things. One, the breadth of neutralization, and then two, the potency of the antibody. So the breadth of neutralization estimates how many viruses out of the 12 are neutralized, while the potency estimates the amount of antibody required to neutralize a particular virus isolate. So to derive our neutralization BP score, we log transform average percent neutralization. A neutralization BP score of 0 means no neutralization, and a score of 1 means complete neutralization of all the 12 viruses. So these parameter, the neutralization BP score, correlates with the commonly used half maximal inhibitory concentration, IC50. And using this method, it allows us to evaluate percent neutralization at a single dilution of 1 to 50. So prior to ART, people with HIV and active tuberculosis as a group had a higher neutralization BP score as compared to people with HIV, no tuberculosis. So in figure here, the neutralization BP score is on the y-axis, and then the disease groups are on the x-axis. So each dot represents a participant. The blue circles are the people with HIV and active tuberculosis, while the red squares are the people with HIV, no tuberculosis. So the heat map in figure B shows different plasma virus combinations with the green color pattern representing plasma virus combination with the highest neutralization values, and the light gray pattern represent plasma virus combination with lowest neutralization values. Disease groups are on the y-axis here, and the 12 viruses that were used in this study are on the x-axis. So the color histogram here shows here with the green, lime green, peach, and light gray representing different ranges of percent neutralization. And then the bootstrap attached to the disease groups on the y-axis group plasma antibody combination with the highest values in cluster 1 as compared to cluster 2 and cluster 3. In cluster 1, most of the plasma virus combination are from the people with HIV and active tuberculosis. So the color and symbol representation are consistent for pre-treatment groups for the rest of my talk. So we next examined antibodies isolated from individuals who received antiretroviral treatment. And then what we did was to just assess the antibodies isolated from these participants after 6 months of treatment. And then we use these antibodies to neutralize all the 12 viruses. So in figure A and B, disease groups are on the x-axis. The neutralization BP score is on the y-axis. And in figure B, the fold increase or change in neutralization BP score is on the y-axis. And then the fold change is the ratio of the post-ART to pre-ART neutralization BP score. So each dot will represent everyone among the different disease groups. The burn cycles are the people with HIV with treated tuberculosis. And the blue cycles are the people with HIV and active tuberculosis. The red squares are the people with HIV with no tuberculosis disease. So across groups, individuals with suppressed virus levels or less than 1,000 copies per milliliter of plasma are represented with colors and symbols with black boundaries. So in figure A, we found no significant difference in the neutralization BP score among all the three disease groups. However, in figure B, the fold increase in neutralization BP score among all the three groups was found to be highest in people with HIV and active tuberculosis compared to both people with HIV, treated tuberculosis, and people with HIV, no tuberculosis. Furthermore, the heat map and bootstrap analysis comparing different antibody virus combinations group most people with HIV with either active tuberculosis or treated tuberculosis in cluster 1. Again, the color and symbol representation are consistent for post-treatment groups for the rest of my talk. So the second function of antibodies that we assessed was antibodies' ability to clear HIV-infected cells in a process known as antibody-dependent cellular cytotoxicity, or ADCC. Similar to neutralization, we employed a cell reporter assay. And in this assay, the MT4 cells engineered to express CCR5 and luciferase were infected with a particular strain of HIV. And after the infection has been established, the relative luciferase expression is at least 10 times higher than in uninfected MT4 cells. So after that, we code these infected cells with each participant antibodies and then introduce natural killer cells, which will bind the FC portion of the infected MT4 cells bound antibodies. And then once these MT4 antibody and natural killer cell complex is formed, the natural killer cells we release granzyme and perforin molecules to clear these infected cells. This will then result in a reduction in luciferase expression as compared to when no plasma or antibodies is added. So we did this for every plasma or antibody sample obtained from participant before and after ART against MT4 cells infected with different HIV strains. Like what we did for percent neutralization, we log transform of the average percent ADCC to score the ADCC breadth and potency, or BP. And again, the ADCC BP score of 0 means no ADCC for all the 10 viruses, and a score of 1 means complete ADCC for all the viruses we used in this study. So in figure A, before ART, we found no significant difference in the ADCC BP score among people with HIV active tuberculosis and people with HIV no tuberculosis. In figure B, after the initiation of ART, we found ADCC to be the highest in people with HIV individuals who develop tuberculosis disease as compared to people with HIV treated tuberculosis, and people with HIV no tuberculosis. And in figure C, the fold increase or the ratio of post-ART to per-ART ADCC BP score was found to be highest in people with HIV active tuberculosis as compared to other disease groups. So we next wanted to know whether the ADCC BP score was associated with neutralization BP score before and after ART. This association would suggest that it is very likely that the same set of antibodies were responsible for neutralization and ADCC in this study. So in figure A, before ART, we found ADCC BP score and neutralization BP score to be directly and significantly correlated. In figure B, we also observe a direct correlation between ADCC BP score and neutralization BP score. However, this correlation was not statistically significant. So in summary, we have shown that tuberculosis disease is associated with the enhancement of neutralization activity before and after the start of ART. We have also shown that tuberculosis disease is also associated with enhanced antibody effector function, which is ADCC, after ART in people with HIV. And we also found that antibody effector function and neutralization activity to be correlated before and after the start of ART. This is suggesting that the same set of antibodies are likely responsible for ADCC and neutralization in this study. So after we established that the specific antibodies in people with HIV and active tuberculosis as compared to people with HIV no tuberculosis were broader and more potent, we next asked whether cytokines responsible for B cell development are elevated in these individuals. So we specifically looked at cytokines involved in B cell development. BAF and APRI molecules can be redundant, but because they have a mutual receptor, in this case, tag high, and overlapping functions. But both cytokines promote B cell survival and class switching. On the other hand, the IL-6 molecules potentially secreted by macrophages in the presence of tuberculosis may promote native CD4 T cells differentiation into T follicular helper cells. A T follicular helper cells and a cognate B cells form a germinal center in the B follicle. So in our studies, we found IL-6, BAF, and APRI molecules to be elevated in people with HIV active tuberculosis as compared to people with HIV no tuberculosis before treatment in figure A to C. After treatment, we still found both BAF and APRI molecules to be elevated in people with HIV active tuberculosis as compared to other groups in figure D and E. In univariate correlation analysis, we found IL-6 and BAF molecule to be directly associated with neutralization BP score before treatment in figures A and B. And after treatment, we again found APRI molecule to be directly associated with a fold increase in neutralization BP score. So in summary, we have shown that tuberculosis disease or active tuberculosis is associated with the elevation of cytokines involved in B cell development before and after ART. Furthermore, these cytokines are associated with the recorded HIV-specific neutralization burden and potency for the same participant, suggesting that it is very likely that active tuberculosis is elevating IL-6, BAFF, and lipid molecules to enhance antibody responses. So based on longitudinal and cross-sectional studies, several determinants or factors that are associated with the emergence of broad and potent neutralizing antibodies in people with HIV have been identified. These factors include high viral load or envelope diversity, which describe the quantity and the quality of HIV antigens. And also we have high HIV IgG and B-cell receptor diversity, and high frequency of T follicular helper cells. All these factors are known to be directly involved in the development of broadly neutralizing antibodies. But in this talk, and because of our time, I think I would discuss the viral load and HIV envelope diversity. So returning to our model, we have shown that B-cell development cytokines are elevated, and that HIV-specific antibodies in people with HIV and active tuberculosis are broad and potent. What we next did was to examine the qualities and quantity of circulating viruses or antigen in these individuals. The reason why we did this was because higher viral load and more diverse HIV viruses are virus determinants of broad and potent neutralizing antibodies. And then coincidentally, tuberculosis disease increased viral load and diversity. So in our study, we did not find viral load to be different before and after treatment among all disease groups. Also, viral load did not associate with neutralization BP score before and after ART. So we next assessed HIV envelope diversity in people with HIV active tuberculosis and people with HIV no tuberculosis. And this is before ART initiation. So what we did here was to isolate total RNA from blood samples, and then we made HIV envelope complementary cDNA, complementary DNA or cDNA, using reverse transcriptions with subtype C reverse primer. And from the complementary DNA library, we amplified the envelope gene using single genome amplification, which is essentially a nested PCR. So this nested PCR involves two rounds of amplification. First, primers were used to bind and amplify the region before HIV envelope segment. And in the second round, primers bind the near HIV envelope gene. And then the PCR product confirmed in 1% agarose gel was then sent for sequencing. So using this method, we were able to generate over 300 sequences, and we assembled them using HIV Align tool. So the first thing we did with these envelope sequences was to construct a rooted phylogenetic tree to show evolutionary relationship among all sequences using the maximum likelihood function. So the maximum likelihood function groups sequences based on regions of homology. So here, external nodes are the ancestral, internal nodes are the ancestral sequences from which the external nodes are believed to evolve from. And the length of the branches show the evolutionary distance between the ancestor and the isolated sequence. So as expected, our sequence is isolated from any individual clustered together. However, there are sequences from 2 people with HIV and no tuberculosis in the cycle that cluster together, suggesting a possible virus transfer between these individuals. So we next estimated a pairwise genetic diversity of sequences for each participant. So the pairwise genetic diversity estimates the average nucleotide substitution rate. Briefly, assuming the substitution rate for A, C, T, and G are clearly defined, the algorithm will calculate the substitution rate along the entire sequence. In this case, sequence 1 versus sequence 2, and do the same for the rest of the sequences in a pairwise manner to score the average genetic pairwise distance. So when we compare the average genetic pairwise genetic diversity among the 2 disease groups before treatment, we found no difference in figure A. Similarly, these average pairwise genetic diversity did not associate with neutralization BP score and ADCC BP score in figure B and C, respectively. So in summary, virus level and diversity is not determinant of broad and potent neutralization activity in this study. The next question we had was whether predicted envelopes sequence motifs from isolated sequences are different in people with active tuberculosis as compared to people with HIV, no tuberculosis. This is because depending on the quality and specificity of antibodies in people with HIV, viruses will escape using strategies other than nucleotide or DNA mutation that I discussed earlier. So here is a structure of HIV envelope with a trimeric, here's a structure of HIV with a trimeric glycoprotein or envelope. And this envelope has 2 subunits, the surface glycoprotein or GP120 and the transmembrane GP41. And as compared to the transmembrane subunit, the surface subunit has a larger surface area and more antibody contact site. So usually we have more broad and potent neutralizing antibodies targeting the GP120 subunit as compared to the ones targeting the GP41 subunit. So now let's look at the electron microscopy structure of the same envelope. Different antibody constant sites are highlighted in red, blue, and other colors with corresponding antibodies. So what HIV does structurally to evade antibody responses is to mask this conserved antibody constant site with glycans or sugars. So these glycans occur mostly on asparagine amino acid. Thus, we have the asparagine or end-linked glycans. Some other glycans occur on serine and threonine amino acid and are dubbed the O-linked glycans. So using the glycosylation or process of adding glycans, HIV is able to mask different antibody contact sites and evade antibody responses. So in addition to glycosylation, other amino acids in different antibody contact sites have also been identified. So what you see in figure A to C, the amino acid residues or logos at different antibody contact sites. The logo height represent amino acid frequencies, while amino acids in red and blue are associated with resistance and sensitivity to HIV-specific antibodies. Amino acids shown in green differ for different neutralizing antibodies, and all represent asparagine amino acid in the potential N-linked glycosylation motif. The contact regions in surface subunit of the HIV envelope, including variable 3, V2, and CD4 are shown in figure A to C, respectively. And based on this parameter, different antibody contact site and sensitivity signature have been described. So we translated all the isolated nucleotide sequences into amino acids and probed for some of the signature sites that I discussed earlier. And in this table, we have the amino acid position based on the reference residue, which is the HXB2. And in the next column, the particular amino acid associated with neutralization were identified and denoted with one letter abbreviation, and whether their presence associated with resistance or sensitivity to antibodies. So the next column here shows the odd ratio. Any odd ratio greater than 1 implies that this specific amino acid at the specified location occurs more in sequences obtained from people with HIV active tuberculosis than in people with HIV no tuberculosis. Lastly, the p-value to show the significance, the statistical significance. So of the 7 amino acids associated with resistance, we found 4 sites, mostly in the GP120 region or the surface subunit of the HIV envelope, to be enriched or more common in people with HIV active tuberculosis as compared to 3 sites in people with HIV no tuberculosis. So in this table, the amino acid hen at the position 293 is a potential N-linked glycosylation site, and occurred more in people with HIV active tuberculosis, suggesting that envelope or circulating viruses in these individuals have the capacity to mask conserved regions with glycans. I think because of our time, I think we're running out of time. I'll just briefly move on to the summary of the study. So based on these, based on everything I said, in conclusion, our study has actually established 3 consequences of active tuberculosis co-infection in people with HIV. One is B-cell development cytokines, including IL-6, BAV, and APRIL molecules are elevated in people with HIV and active tuberculosis. And HIV-specific antibody responses are elevated in the presence of tuberculosis disease. And the third consequence is that viruses in people with HIV and active tuberculosis are predicted to be escaping from GP120 targeting antibodies. And our study is significant for the following reason. The development of broad and potent neutralizing antibody is a rare event occurring in about 1% of people with HIV. Attempt to induce broad and potent neutralizing antibodies in humans have been unsuccessful. And the third reason why this study is significant is that we found that the development of broad and potent neutralizing antibodies was more common in the presence of active tuberculosis. However, this study is not advocating for Mycobacterium tuberculosis co-infection as a means to boost antibody responses in people with HIV. This is because tuberculosis disease is bad and even worse with the HIV co-infection. TB worsen HIV disease progression, and conversely, HIV reactivate latent TB infection. But I hope that after we establish mechanisms used by tuberculosis disease to enhance antibody response in people with HIV, we can apply these mechanisms as part of vaccine regimen, potentially agonists that can trigger pattern recognition receptors pathway involved in tuberculosis-associated antibody perturbation. This approach may be used to boost vaccine-induced antibody responses. And yeah, I would like to acknowledge important people pivotal to this project that have contributed immensely to this project. And yeah, I'll take questions.
Thank you very much, Bukola, for such a wonderful presentation. It was really nice to have you take us through the journey of your research, and it was so pleasant to the ears, and there was so much to learn. So I would like to take this opportunity to call on everyone for your questions. So we are ready to receive your questions. If you would like to ask your question verbally, you can please raise up. You can raise up your virtual hand, and you'll be called upon to unmute and ask your question directly. And if you would prefer, you could put down your question in the chat box, and it will be read out on your behalf. So let's avoid the awkward silences. Let's have your questions, please. So to start with, Dr. Bukola, to be honest, I think your disclaimer it took away my question, really, because I was going to ask about, what were the implications of these findings for clinical, what might they pose for clinical treatments. But in a way, I would imagine that the disclaimer perfectly really puts it in view as regards, okay, we wouldn't say, okay, let's co-infect people, intentionally co-infect people with HIV just to get more broadly neutralizing antibodies. But possibly that's an area to just look at more in vitro, to really understand the mechanism and see how we can replicate the same impact that active Mycobacterium might have on the effect might have on the HIV. So I'll be taking a moment to look through the charts. All I can see is thanks. Yeah. I also echo the thanks. Thank you so much for such a brilliant presentation. So
thank you.
Do we have any questions, guys? I might be tempted to call names. Akinloye, what was yours?
Hi, hello.
Can you hear
me? Yeah.
Yeah, thank you so much, Dr. Bukola, for the presentation. It was really insightful, and despite the fact that I missed some parts of the presentation, I was still able to get a lot from it. Now, despite the disclaimer, I think I still have 2 questions that I have here. Well, it's one of it is not like a question. It's just like just to get your opinion with respect to the outcome of the whole of this project. Now, if you take a very close look at the outcome and the experimentation design and everything, notice that there are differences between pre and post AR3 treatments. The outcome that's the outcomes that we got from pre and post. From your own opinion, does that mean we are nowhere near the end of the use of ART, despite the likely implementation of this treatment approach? Does it mean that because apparently the influence of antiretroviral treatment is also there. So what's your opinion with respect to that? So that's the first one. So the second part of the question is the influence of viral load. I didn't get, I think that part of the presentation that I missed, the influence of viral load on the neutralizing antibodies. Now, fine, maybe you design an experiment, to get that influence, but in the patients, or in the participant that participated in the study, did you take their viral load before treatment and after the treatment? I mean, to be able to compare the viral load in that context, or was that exactly what you did with respect to the influence of viral load? And that's the second one. So another third question came to my head now. These patients, were they also taking anti-tuberculosis drug? Because I don't think you mentioned that throughout I'm not certain whether I missed it. So was it only antiretroviral drug they were taking? Because of course, I mean, people with Mycobacterium, tuberculosis infection, they will have to be taking anti-tuberculosis drug alongside. And okay, yeah, so the fourth question is actually related to the viral load thing, because my perception is usually the manifestation of HIV symptoms, it's kind of dependent on the viral load, to the best of my knowledge. I mean, the main goal of antiretroviral drug is to suppress the viral load. So I believe the release of whatever antibody should be dependent on quantity, or maybe I also missed that part of the presentation. Thank you. Sorry if you had discussed any part that I'm bringing you back on. I'm sorry about that.
No worries at all. Yeah, those are great questions. Honestly, those are like fantastic questions. So I think to your first question, which was what do I think about ART? Is it here to stay, or are we at the end of ART? I would say. I would say I think for now ART is here to stay.
Okay.
The way I see vaccine, HIV vaccination program, the future will be to essentially assess regions and then see what are the circulating HIV variants, say in West Africa, and then try to find antibodies that are known to be able to neutralize those viruses that are circulating in a particular region. So you want to actually compare the circulating viruses and also look at your antibody inventory and see, among all these broad neutralizing antibodies, which one can actually neutralize all these viruses. So the approach will be a regional vaccination program, in the sense that we have to account for the circulating variant and then develop our vaccine based on that. I don't see any fix-it-all vaccine for now, because if you look at a study, let me go back in time. Yeah, this study, this was a big study, and in this case, only the viruses that the antibody is sensitive to, that's where you see significant improvement. That is to say, about 75% efficacy of the antibody treatment. So that's a big problem. And also you also have to take a look at what is the antibody decay time or half-life. So you want to consider a lot of factors. So that's why I think ART is here to stay. I would say
Okay.
there are currently a lot of improvement on ART in the sense that we now have long-acting ART that you only need to take maybe once in a month, and they'll be long-acting. So that's makes everything easy for as part as in the sense that people are able to comply with medication and prescription. Yeah. So to your second question, which was, did we examine viral load before and after ART? Yes, we did. We actually checked the viral load before and after ART. And as you would expect, the viral load was suppressed after the initiation of ART, mostly in all the individuals. There are cases where individuals were unable to reach suppression, probably because they did not comply with their medication, also, or probably because they have viruses that are escaping. I most likely think it's because they did not take their prescription. And then the third question was there tuberculosis treatment? Yes, there was. These individuals were treated based on their national guidelines. We had samples from Uganda, and I think for Uganda, in Uganda, you have to like take tuberculosis antibiotics for 6 months before you even start antiretroviral treatment. So that's the national guideline. So like based on the national guidelines, people were treated accordingly. I think your third question was your fourth. What was your last question, Eva? About viral
I think
load?
it's just to the last question is to kind of come to a conclusion with respect to the viral load observed in the patients and the viral load. I mean, the influence of the viral load to the neutralizing antibody. I mean, I think I got a glimpse of a slide that there was no influence of the viral load, right, on the neutralizing antibody. So, I mean, bringing that to the changes in viral load that were observed in the main patient, the patient that the study. I'm trying to find the
Oh yeah.
match.
Oh yeah. I clearly understand that. Yeah. So here's a principle in the feed. It is generally believed that high viral load would translate to more antigen and even diverse antigen. And so when you have this increase in diversity of antigen, you're able to broaden your antibody responses in the sense that from recognizing 3 different variants, you're able to recognize 100. So the idea is that with high viral load and higher or envelope diversity, you will eventually produce broadly neutralizing antibodies. Yeah, but in our study, we didn't see that to be the case. Instead, what we saw was that there appears to be means beyond the nucleotide levels. That's the amino acid motifs that we actually saw to be completely different. So what we saw was that viruses in people with HIV, active tuberculosis, actually have envelopes, amino acid signatures that suggest that they're able to evade antibody responses using mechanisms that are not well looked at, which include the ability to cover your envelope with glycans, because if you look at the cell, every cell in the human body has glycan. So it's so difficult for the antibody to differentiate between HIV and human cell in that case. Yeah.
Okay. Yeah. Thank you.
Yeah,
Thanks so much. Yeah, well done.
All right. Thank you very much. In the absence of further questions, on behalf of the Science Cafe community, I'd like to thank you, Dr. Bukola Adeoye, for taking the time and for sharing your research data with us in such an insightful session. Thank you very much. At this point, the session will be coming to a close. I'd like to thank everyone for joining us. But briefly, I'd like to invite Martins or Wells if there are any announcements, please.
Potent agonist for different toll-like receptors, which is the same way the latency referencing agent practically works, because you're just activating the cells and asking the cells to make more viruses, and after that you're able to eliminate them, especially in the context of ART. So once we learn more about how tuberculosis is actually perturbing immune system in the context of HIV, we'll have more answer on that. Another thing I would like us to think about is that HIV transcription actually affect our antibody responses. It's so possible that in people with ART, we can always have this low level of transcription to enhance antibody levels, in the sense that although people are taking ART, if we understand how tuberculosis is activating immune cells, we can also mimic the same thing in people with HIV and ART. Essentially, we are making more antigen, although they will be low level, but at the same time, they'll be more diverse in the context of ART. And there are a lot of things that happens with the antibody conjugation when you have HIV infected cells, and then these cells are actively expressing HIV, and then you can always count on antibodies to recruit natural killer cells, essentially the antibody dependent cellular cytotoxicity that I discussed earlier, which is, as we may know, ART cannot penetrate some sanctuary sites. So I feel like this is going to be very, very important if we want to actually reduce latency or latent reservoir in people with HIV. We want to look at these things holistically and see what would be the overall advantage or benefit if we're able to boost immune responses like the way tuberculosis would. That essentially will be answered after we have a model that can be used to study these co-infection, and also sort of influence these different conditions with this co-infection. I'm not sure if I answered your question, because I think I talked too much. So yeah.
Yeah, you did. Thank you so much.
Okay.
I think I have another question, please.
Okay.
Okay, so I wasn't around when you talked about the broadly neutralizing antibodies. So I just want to confirm if they are effective across multiple viruses or bacteria, like multiple infectious diseases, or it's just limited to HIV and TB. Then my second question is, okay, I can go ahead.
Yeah, so to your question, these viruses, these antibodies that we're talking about are only, they only work against HIV variant, not against tuberculosis or any other pathogen.
Okay.
Yeah.
Okay, so another question is, I learned that interferon gamma is quite effective against tuberculosis. So I'm wondering, since you are studying HIV and tuberculosis co-infection, why it wasn't maybe one of the cytokines that you assayed for?
Oh, so so for this study, we actually only concentrated on the antibody responses. That's why we kind of like selected cytokines that are known to improve antibody responses. So, of course, you're going to have elevation of a lot of cytokines, because that's what I think happens in the context of active tuberculosis, because you have like this broad activation and you have a lot of cytokines being upregulated. But we only concentrated on cytokines that are known to improve antibody responses. That's why.
Okay, thank you so much. That's all.
Yeah.
Thank you.
So, thank you so much, Walt, for the questions you asked, and as well, thank you, Dr. Bukola Adeoye, for doing justice to those questions. So once again, I'd like to thank you for the presentation, and I'd like to thank everyone as well for attending. So this brings this session to a close. Thank you guys, and enjoy the rest of your weekend. Bye.
Bye-bye.